The function of the helper T cells. These helper T cells share some common features with that of the cytotoxic T cells, but they also differ in some respects. Here, the helper T cells and the cytotoxic T cells share the following common feature. These common features are their TCRs are made up of alpha and the beta chain. The cytoplasmic tail of the TCR is associated with the CD3 complex having the items, and they have the co-receptor associated with the LCK. And whereas they differ in the following respect that the co-receptor in the helper T cell is CD4 instead of the CD8, and and their TCR recognize the foreign peptide MSC class second protein only. And this CD4 is made up of the single polypeptide chain and has a four domains, that is D1, D2, D3 and D4. Here the structure of this CD4 domains having the different and four domains having a D1, D2 and D3 and D4 domains, and another is the antigen presenting cells. These are the antigen presenting cells as the MHC II proteins are present in the macrophages, follicular dendritic cells, and the B lymphocyte. These cells are known as antigen presenting cells, and these are very much important for the um, action of the MSC class second. Um, Here, these are the dendritic cells which are present all over our body, macrophages, and the beta B lymphocytes. The TCR of the helper T cell bind to an antigen fragment displayed by the MSC class second protein of the APC. The D1 domain of the CD4 bind to the B1 and domain of the MSC class protein, MSC class second protein. Here the D1 is binding to the beta 1 domain. Okay, after this binding, the intracellular FYN is activated mm, by the CD45, and FYN phosphorylates item of the CD3 complex, and phosphorylated items are docking site for the JAP70. This JAP70 bind to the phosphorylated items and phosphorylated by the LCK. The JAP70 phosphorylate the tyrosine residue of some target proteins, and this result in release of the cytokines from the helper T cells. Here is the representation of the action of the helper T cells. It is taking place through the help of the MSC class second proteins. Antigen fragment is present in between the beta 1 and the alpha 1 regions. Here the another is the T cell receptor having the alpha 1, alpha 2, beta 1 and beta 2 region along with this gamma, delta, epsilon and zeta chains, and these in this the co-receptors here is the having the CD4, this having the four domains D1, D2, D3 and D4, and this and the other phosphorylation process going on, and this helps in the action of the class second uh, MSC proteins. Then these lymphokines, that is the cytokines that are released by the helper T cells, includes the IL-2, IL-3, IL-4, IL-5 and IL-6, granulocyte colony stimulating factor, that is GCSF, and granulocytes macrophages colony stimulating factor GM-CSF, and then tumor necrosis factor (TNF) and the interferons gamma (IFN gamma). And the action of the lymphokines is such as this: the interleukin 2 stimulate the multiplication of the helper T cells themselves, and interleukin 3 stimulate the proliferation of the bone marrow stem cells. Interleukin 4 stimulate the proliferation of the B cells and the T cells and the mast cells. And whereas the interleukin 5 stimulate the growth of the eosinophils. Interleukin 10 stimulates and synthesis of the MHC class II protein in the B cells, whereas the GM-CSF stimulate the growth of the granulocytes and the macrophages. IFN gamma stimulates and activates the macrophages and the natural killer cells, and the TNF activates the macrophages. And some of the lymphokines and that attracts the phagocyte cells to the site of an infection, and thus the release of these cytokines lead to the various cascade steps, and this leads in the increase in the number of the helper T cells, increasing in the number of the cytotoxic T cells, increase in the number of B cells and the plasma cells, and increase in the number of antigen presenting cells, and increase in number of the phagocytic cells, and the activation of those of the phagocytic cells, and thus the both innate and the adaptive immunity are generated up to the fight against the pathogens, and the co-coordinated and effective immune response is mounted against the pathogens. The helper T cell respond to call for the help. The call is given by the antigen presenting cells, such as dendritic cell macrophages. This call is in the form of an antigen fragment displayed by the MHC proteins. These helper T cells have no direct role in destroying the antigens. They activate different arm of defense systems and then coordinate their actions. The helper T cells are described as the commanding officer of the defense force. If the helper T cells are depleted or in less in amount, then the entire human immune system is destroyed. That is prepared. Then some of the function of the suppressor T cells, or these are the regulatory T cells and that possesses the TCR and the CD8 that plays the regulatory role in the immune response. Shut down the immune response after the invading organism has been destroyed, and this is very much requirement to suppress the immune system. Otherwise, the inflammation and the other things can happen inside the body continuously. Suppressor T cells release some of the lymphokines after the immune response has been achieved its goal. This this signals all other immune system participate to cease their attack. And the suppressor T cells also prevent immune response against the self